How's it going, folks? Happy Monday. You guys hear me okay? All right. It is 3:01, so let's get started. And so I'm sure all of you have seen, I did grade exams last week. And if you haven't had a chance to take a look at the exam, please go over your exam carefully. Okay? Go over question by question. If you do have any questions, any doubts, any issues, please send me an email with a question number and I can take a look at it. A couple of things that I want to say about that, though. Do not look at the marking of the exam because the exams are graded automatically. And then I have to go over manually through the exam and then make the corrections that needs to be made. So don't look at the markings, look at how many points you got for each one of the questions. Okay, that is important. Also, I had questions about this before in my previous class. For a bone marking question, if you got the name of the marking wrong, there is no partial credit. Okay, so there's no partial credit if you got the name of the marking wrong. If you got the name of the marking correct, but then you missed the name of the bone, then there is partial credit for that. Okay. How was the exam? It was a mixed bag. I'll be honest with you. The highest score was a 48. The lowest score was a 14.5, and then we had everything in between. Okay. Um, and the thing, though, is that I kept telling you folks from the very first day of class, which was 7 weeks ago, right? 7 weeks ago, almost 2 months before we had our first exam. And I told you folks several times, you need to start to study. There's a lot of terms of bones and muscles and bone markings that you need to memorize. And many of you did not do that. And that I can see that on the result of the exam. Okay. The good news is that the first exam is done and the next two exams are easier. Pay attention. I'm not saying that they are easy, but they are definitely easier than the first exam. All right. The bad news is that you don't have 7 weeks anymore to prepare for the next exam. The next exam is going to be coming up pretty soon. And you need to start looking at those exam lists, folks. You have them. Look, you guys, usually students beg for a study guide, right? I get this every class, every semester. Professor, where is the study guide? Give me a study guide. You have a study guide from the first day of class. Whatever is in the list, that's what you're responsible for. So you don't have to worry about guessing what's going to be on the exam, what's going to be included, what's not going to be included. It's already there, spelled out in the exam list. All you have to do is open the list and start studying for the exam. Okay, and you need to start doing that today, now, because again, the next exam is going to be coming up pretty soon. And the format of the exam is going to be exactly the same. You're going to have 50 questions. You're going to have pictures of models, and you need to answer, put in the name of the structure that you're looking at. All right. Any questions, folks? Anything at all about the first exam? And we're going to do better in the second exam. It's okay. You can lie to me. That's fine. All right. So today we're already going to go over and take a look at some of the structures which we're going to be looking at for the second exam. In today's class, we're going to talk about the nervous system. And for the folks that are in my lecture, this is not going to be something new. We have already seen it. And I want to go over the physiology first, right? I want to talk about the function of the nervous system, the different parts of the nervous system. And then I want to spend some time talking about the anatomy, especially focusing on the sections of the brain, the parts of the brain that will be on the exam, on our next exam. All right. Now let's take a look at the big picture first. Let's understand, why do we have a nervous system? What are the main parts of the nervous system? And the function of our nervous system, folks, is that our nervous system help us to respond to changes in the environment. Now, when I say environment, I'm not just talking about the weather outside, which have been very hot lately, right? No, I'm not just talking about that, but I'm also talking about the internal environment, the things that are going on inside of our body. And both outside of our body and as well as inside of our body, we have a lot of receptors. And these receptors, what do? Receptors perceive stimuli, whatever that stimuli might be. Let me give you some examples of stimuli. Light, sound, smell, taste, touch, temperature, texture, levels of glucose in the blood, the levels of oxygen in the blood, the levels of CO2 in the blood. These are all different stimuli. And the receptors will perceive the stimuli, and then they're going to send that information up the ladder through a group of neurons called the sensory neurons, and that information is going to go to the brain. And the pathway that goes from the receptors to the brain, and this is what we call the afferent pathway. Now, once that information gets to the brain, what is the brain going to do with that information, folks? The brain is going to decide what to do, if anything at all. Now, once the brain makes the decision, the brain now is going to send that decision, whatever the decision might be, down the line through what we call the motor neurons, which is a different group of neurons, through a pathway called the afferent pathway. And that information now is going to go all the way down to the effectors. And the effectors are the ones that actually do the work. Not the brain. The brain is the boss, right? The boss don't do any work. It's actually the effectors which are going to decide, which we are going to actually do the work. Let me give you an example of effectors. Different endocrine glands in our body, our skeletal muscle. These are all different effectors. Okay? Now, the nervous system is divided into several different sections. It is divided into what we call the sensory neurons, and we have the motor neurons. And when we get to the motor neuron division right here, this is going to be further subdivided into two smaller teams, two smaller subdivisions. The motor neuron is going to be subdivided into what we call the SNS, the somatic nervous system, and the ANS, the autonomic nervous system. The SNS, the somatic nervous system, this is the neurons, the nervous system that is controlling our voluntary muscles, skeletal muscle. The ANS, these are the group of neurons that are controlling our involuntary muscles, like the smooth muscle and the cardiac muscle. And then when we get to the ANS, this is going to be even further subdivided into what we call the sympathetic and the parasympathetic divisions. But you don't need to worry about that right now. Okay? Now let's take a look at and see what a neuron looks like. And a neuron is a very special cell in many different ways, folks. One of the things that is special about a neuron is the fact that neurons are quite big. They're big cells. And another unique thing about neurons is that neurons can become excitable. They can become excited. What does excited mean? Excitable. It means that they can generate action potential, just like the skeletal muscle cells. Skeletal muscle cells can also become excitable. Okay, so these are two things that are unique to both the skeletal muscle cells and neurons. Now let's take a closer look at the different parts of the neuron right here. And a neuron kind of look like my arms and my fingers and my hands right here. So imagine that this what you're looking at right now is a neuron. And the palm of my hand would be what we call the cell body of that neuron. And in the cell body, we're going to find the nucleus, we're going to find organelles, we're going to find proteins, all the different parts that you usually find in a cell. Now, attached to the cell body, we have these finger-like structures. These are what we call the dendrites. And the dendrites, they receive information from other neurons. And then on the opposite end of the dendrites, we have this bigger section right here, this long section. This is what we call an axon. And at the end of the axon, we're going to have what is called the axon terminals. Okay? And the action potential, the flow of information that is passing through the neuron, it goes in one direction. What direction is that? It's going to go from the dendrites, it's going to pass through the dendrites, pass through the cell body, run through the axon, and then they're going to arrive at the axon terminals. And at the axon terminals, the action potential is going to lead to the release of neurotransmitters into the synapse, which is going to allow the propagation of the action potential. Okay? Now, we can classify neurons based on the function of that neuron and also based on the structure of the neuron. Within the function of neurons, we have three different types. What are they? We have what we call the sensory neurons, we have the interneurons, and we have the motor neurons. Okay, again, following the pathway of the flow of the information through the nervous system. Now, in terms of the structure of the neuron, they can be classified as multipolar, bipolar, and unipolar, depending on how many processes are attached to the cell body. For example, this neuron right here would be a multipolar neuron because it has several dendrites and an axon attached to the cell body. So it has several processes, okay? Now, as I said earlier, muscle fibers, muscle cells, and neurons, they can become excitable. And what that means, it means that they can generate an electrical impulse that we call action potential. And both of these different types of cells here, both the skeletal muscle and the neurons, they have a resting state. You know, sometimes they're not doing anything, they're sleeping. If a muscle cell is not contracted, it is resting. If a neuron is not receiving any stimuli, it is resting. And when those cells are at rest, inside of the cell is negatively charged, while outside of the cell is positively charged, right across the membrane. In fact, for a neuron, what we call the resting membrane potential is minus 70 millivolts. So it's negatively charged, okay? Now, what maintains this resting membrane potential of both a muscle fiber and a neuron? This guy here maintains that, folks. This little pump called the sodium-potassium pump. And the sodium-potassium pump is constantly pumping 3 sodiums outside of the cell and 2 potassium inside of the cell. Now, we can see that we have a 3 versus 2 being pumped, 3 outside, 2 inside. So which means that outside of the cell is going to be slightly more positive than inside of the cell. And it's very important to maintain all of the sodium outside of the cell. Why? Because the sodium is going to create what we call a chemiosmotic gradient, in which we're going to have this huge, massive concentration of sodium outside of the cell and a smaller concentration of sodium inside of the cell. Now, is this in balance when we have all this sodium outside and very little sodium inside? No, this is not balance. This is not homeostasis. So the sodium wants to get back inside of the cell, okay, to be in balance. However, the sodium cannot go through the phospholipid bilayer because it is positively charged. And when the cell, the neuron, is at rest, the ion channels are closed, which means that the sodium stay outside, accumulating outside of the cell because the sodium potassium pump keeps pumping sodium outside of the cell. Now, when we have an action potential arriving at the axon terminal of a neuron, that is going to induce the release of little neurotransmitters called acetylcholine. What is the acetylcholine is going to do, guys? The acetylcholine is going to bind to receptors on the ion channel, and they will open up the ion channel. Now that the ion channels are open, we're going to have a flood of sodium inside of the cell, changing inside of the cell from negative to positive and outside of the cell from positive to negative for a very fast little time. Now, this change in membrane polarity, again, from negative to positive inside and positive to negative outside, that will create a spark. And that spark, folks, is action potential. Okay, and this action potential will keep on propagating. And as I mentioned earlier, action potential moves along the neuron in one direction only. It doesn't go back and forth, one direction only. What direction is that? Dendrites, cell body, axon, axon terminal, and when the action potential gets there, it will release neurotransmitters into the synapse. Okay? Which again is going to allow the continuation of the action potential in the neighboring neuron. Now, when a neuron is resting, we say that in the neuron, in the cell is polarized. So when the cell is at rest, the cell is polarized. What does that mean to be polarized? Guys, polarized means that this cell that is at rest, this neuron that is resting, there is energy potential in that cell. Potential, the key word here is potential. Okay, potential. Now, once the cell receives a stimulus and the sodium channels start to open, allowing sodium to go inside of the cell, changing inside of the cell from negative to positive and outside from positive to negative, that generates a new action potential. So now we say that the cell right here in this region that I just circled, the membrane is depolarized. So this is what we call a local depolarization of the membrane. Now it's called the depolarization because now that potential energy has been realized. We have the generation of action potential, of electrical impulse. So what's going to happen now is that we're going to have this constant depolarization and repolarization of the membrane, which will push the action potential forward, going towards the axon terminal. Okay? Now, we can also say that action potential follows what we call the all or none principle. And again, none of the stuff that I'm telling you right now should be new to you. We already covered this in class, in the lecture. Now, what does it mean to be all or none principle? It means that either an action potential will happen or it's not going to happen at all. Depending on what? Depending if the amount of sodium entering the cell will jump over this threshold right here of minus 55 millivolts inside of the cell. If we have enough sodium entering the cell to go over the threshold, action potential will happen all the way, as we can see right here. If there's not enough sodium going inside of the cell to jump over the threshold, action potential will not happen. And the cell is going to go back to the resting membrane potential. Okay? And the last thing I want to talk about here before we move on into and talk about the anatomy and the different parts of the nervous system, let's talk a little bit about reflexes. Now, the one thing we know about reflexes is that they are fast, they are quick. But we do have two different types of reflexes. We have a somatic and we have autonomic reflexes. How do they differ, folks? Somatic reflexes are the reflexes that involve skeletal muscle, voluntary muscles. Autonomic reflexes, these are the reflexes that involve involuntary muscles, like the smooth muscle, like the cardiac muscle. Among the somatic reflexes, we can have what is called a 3-neuron reflex arc or a 2-neuron reflex arc. What is the difference between a 3-neuron versus a 2-neuron? Guys, the number of neurons involved with that reflex. A 3-neuron reflex arc involves the sensory neurons, interneuron, and the motor neuron. The withdrawal reflex is an example of that. The patellar reflex is an example of a 2-neuron reflex arc that only includes the sensory and the motor. There is no interneuron in the middle. Okay, which makes a 2-neuron reflex arc even faster than a 3-neuron reflex arc. Now, what about the autonomic reflex? An example of an autonomic reflex would be what we call the pupillary reflex. Right here. Now, what is the pupil? The pupil is the hole through the iris, and the pupil size is controlled by the iris. And the iris is made of smooth muscle. So the colored part of our eye, folks, is smooth muscle. So when we walk outside and it's very bright outside, what's going to happen to the size of the pupil? The pupil will constrict. It's going to be very tiny. Why? To decrease the amount of light going inside of the retina. Because too much light inside of the eye, folks, can damage the photoreceptors of the retina. This is why people say, and you should listen, never, ever, ever look directly at the sun because you're going to go blind. That's going to damage your photoreceptors. Okay. Now, what happens when we walk into a dark room? Our pupil will dilate. And the pupil is going to dilate to allow more light to enter the eye so we can see better with the low levels of light. Okay. All right, guys. So let's take a look at the brain, and we're going to see what are the parts of the brain, what are the structures of the brain. I will ask you questions about this in our next lab exam. You need to know the different parts of the brain to be able to identify them, to be able to name them. Okay, and all of these right here that I'm going to go over with you is in the exam list. Now, we can divide the brain into 4 different structures. We have the cerebrum, the diencephalon, the brainstem, and the cerebellum. What is the cerebrum? When you look at the brain right here, folks, the surface of the brain, and you can see all these undulations on the surface of the brain, that is the cerebrum. Do we have 2 cerebrums? Yes. We have a right cerebrum and we have a left cerebrum. And they are divided right in the middle by a fissure that we're going to talk more about it in a minute. Okay. Now, if you look all the way inside of the brain, and we're going to take a closer look in the next slides, excuse me, we're going to find another structure called the diencephalon. And the diencephalon is divided into 3 smaller structures that you guys need to know by name. We're going to go over them. Then right inferior to the diencephalon, we're going to find another region of the brain that is called the brainstem, which also has 3 different regions. And by the way, guys, our brain stem is our primordial brain. You know, when during the process of evolution, the first brain that evolved in animal is the brain stem. Because the brain stem, they control our most basic physiological functions. The brain stem controls our breathing. The brain stem controls our cardiac rhythm, the beating of our heart. This is why we can go to bed at night and sleep. We're not going to worry about breathing. We're not going to worry about beating our heart because the brain stem is controlling those functions. Okay? Now, right all the way in the back of the brain, we're going to find another structure that is called the cerebellum. And please, 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 do not confuse cerebrum with cerebellum. It happens a lot in exams and quizzes. Okay. Now, we all know that our brain and the spinal cord, for that matter, they are covered by three layers, three membranes that help to protect the brain and the spinal cord. And collectively, those membranes are what we call the meninges, right? And what are these membranes? We have the outermost layer that is called the dura mater. Move this out of the way. Then we have a layer right underneath the dura mater that we call the arachnoid mater. And then a layer all the way inside of those regions, of those membranes, a layer that is actually touching the brain itself that we call the pia mater. So again, we have a dura mater, arachnoid mater, and pia mater. Now, in between these membranes, in between the meninges, folks, we are going to find spaces. And the spaces are filled with CSF, cerebrospinal fluid. The space between the dura and the arachnoid mater is what we call the subdural space. So let's say someone hits their head, okay? And then they go to the doctor and the doctor is going to do an MRI, do different diagnostic imaging, and they're going to find a bleeding on that space. So this would be what we call a subdural bleeding. So bleeding in the subdural space. Now the space between the arachnoid and the pia now is what we call the subarachnoid space. Sub means under. Okay. Now, why do we have a CSF? One of the main functions of the cerebral spinal fluid is to help absorb shock so that it protects our brain. When we hit our head, that the fluid inside of the cranial cavity is going to help to protect us. Now, the CSF also helps to remove toxins from the brain because the cerebrospinal fluid is circulating, folks. It doesn't stay in one place. It circulates. Now, not only we're going to find CSF, in the subdural and the subarachnoid space, but we're also going to find CSF inside of the brain itself, in a little cavities, little spaces called ventricles. So, and we have a lateral ventricles, which is the first and the second ventricle. We have a third ventricle. We have a fourth ventricle. And connecting the third with the fourth, we have what is called the cerebral aqueduct. And below the fourth ventricle, now we have what we call the central canal of the spinal cord. Now let's take another look at these ventricles right here in this ville. So there we have it. So this is the lateral ventricles, the first and the second. Third ventricle, cerebral aqueduct, and then all the way at the base of the cerebellum, we're going to find the fourth ventricle, and below that, the central canal of the spinal cord. And guys, again, the CSF keeps on flowing, circulating from one point to the other, okay? All right, now let's get back here to the cerebrum and let's see what are the parts, what are the structures that you need to know for the exam, because I will ask you questions about this in the exam. And again, when we look at the surface of the cerebrum, we can see that it's all folded, all invaginated. Why is it folded, guys? It is folded to increase the surface area to allow more neurons within the cerebral cortex. Now, if I take a slice, and I hope everybody's taking a look right here. If I take a slice of the surface of the brain, the cerebrum right here, and I amplify this, that's what I would see here. And we can see all these little bumps, all these undulations, they are called the gyri. So what is the gyri? The gyri are the little bumps on the surface of the cerebrum. Now, gyri is plural. Gyrus, if I'm talking about one of this guy right here, this is singular, okay, a gyrus. Now, we can see that in between the gyri, there are some spaces, some indentations right here. So we can see a shallow indentation, and we can see a deeper indentation right here. These indentations, folks, are what we call the sulci and the fissures. What is the difference between a sulci and a fissure? The sulci are shallow. Now, sulci is plural, sulcus is singular. So if I'm talking about a single one of these, a single shallow indentation between two gyri, this is a sulcus. If I'm talking about several of them, it's a sulci. Now, there are two sulci that you guys need to know for the exam. I will ask you questions about it in the exam. Everybody look up here, please. One of those sulcus is this line right here, separating the frontal lobe from the parietal lobe. This is what we call the central sulcus. The central sulcus. Do we have a two central sulcus? Absolutely, because we have a two cerebrum. We have the right cerebrum, we have the left cerebrum. So we have the right central sulcus, we have the left central sulcus. The other sulcus that I want you guys to know by name is this line right here. Everybody look up. This line right here that is separating the frontal lobe from the parietal and the temporal. This is what we call the lateral sulcus. And again, it is bilateral, right and left. Okay? Now, what about the deeper indentation between two gyri? This is what we call a fissure. And there are two fissures that I want you guys to know by name. So a fissure is a deep sulcus. One of the fissures that I want you guys to know by name is a fissure called longitudinal fissure. Everybody look up here. This is a fissure, folks, that goes right in between the right and the left cerebrum. It goes from anterior all the way to posterior, literally separating the two hemispheres of the brain, right and left. The other fissure I want you guys to know is the fissure that separates the cerebrum from the cerebellum right here. This is what we call the transverse fissure. The transverse fissure right there. Okay? Now, the cerebrum is divided into different lobes, and there are 4 lobes that you guys need to know for the exam. What are those lobes? We're going to find the frontal lobe, the parietal lobe. They are. So we have right frontal lobe, left frontal lobe, right and left parietal, right and left occipital, right and left temporal. Okay, and don't confuse the occipital lobe all the way in the back of the brain with the cerebellum, which is all the way here. Completely different structure. Any questions, guys? All right, so let's take a look now inside of the brain. We just look at the cerebrum. Now I want to take a look at the diencephalon and the brain stem. Okay, do I have a question here? The occipital lobe has two hemispheres. Uh, yes, all of the lobes have two. Okay, all of the lobes are bilateral because we're going to find lobes both on the right cerebrum as well as on the left cerebrum. Okay, any other questions, folks? All right. So here we have a sagittal section of the brain. Actually, to be precise, this is a mid-sagittal section. Can anybody tell me, am I looking at the left or the right cerebrum here? What is this, guys? Left or right? Here.
Left.
Absolutely. That is the left. Okay. So we took the right and we threw it in the trash. We were not looking at it right here. And what I want you guys to notice here is that this entire section, this brownish section right here that I'm circling right now, this is the cerebrum. So this would be the left cerebrum. All right. And the cerebrum here, as I mentioned, they are completely separate from one another, the right and the left, with one exception. And the exception is this structure right here, this lighter colored structure that we call the corpus callosum. What is the corpus callosum? The corpus callosum is literally a bridge, a tissue that is going to connect the two hemispheres of the brain. Guys, I can take a brain. Let's say I have a brain in my hand, okay? And I'm going to put my finger, draw my finger right here. I'm going to put my finger right there into what we call the longitudinal fissure. That's a pretty good finger. So I can put my finger and wiggle the two hemispheres, and my finger is going to go down into the longitudinal fissure until my finger gets to the corpus callosum. Because now I cannot go any further. Cannot go any further. So what is the corpus callosum doing? The corpus callosum is connecting, is bridging the two hemispheres of the brain, allowing for communication between the right hemisphere and the left hemisphere of the brain. Okay? Now let's take a close look at the diencephalon. And the diencephalon is divided into three regions that you guys need to know for the exam. Also, the brainstem is divided into three regions. What are those regions? We have the thalamus. We have in front of the thalamus, we're going to find the hypothalamus. And then we have behind the thalamus, we have the epithalamus. So again, what are the three regions of the diencephalon? Thalamus, hypothalamus, epithalamus. Hanging from the hypothalamus, folks, we have this very important endocrine gland right here that we call the pituitary gland. We're going to talk a lot more about the pituitary gland later. Again, the pituitary gland is part of the hypothalamus. In the epithalamus, we're going to find another gland, another endocrine gland called the pineal gland. Okay, and the pineal gland produces a hormone called the melatonin. And the pineal gland is part of the epithalamus, like the pituitary gland is part of the hypothalamus. Now let's move on to the brain stem. The brain stem is also divided into three different regions. We have the pons. Sorry, we have the midbrain right there, which is the most superior section. Underneath the midbrain, we're going to find the pons. And underneath the pons, we're going to find the medulla oblongata. Again, midbrain, pons, medulla oblongata. What do we find below the medulla oblongata? Below the medulla oblongata, guys, is going to be the spinal cord. So the spinal cord starts right there, and it keeps going down all the way to the vertebral column. Okay, any questions, folks? FYI, I want you guys to write this down, please. The structures of the diencephalon and the structures of the brainstem are not bilateral. Again, the diencephalon structures and the brainstem structures are not bilateral. Okay, they're just one single structure. Moving on, we all know that the brain is formed, and the spinal cord as well, for that matter, they're both formed by two different matters. They are formed by what we call the gray matter, and we have the white matter of the brain. We have talked about this before in the lecture. Why is the gray matter grayish? The gray matter, which we can find right here in what we call the cerebral cortex. The gray matter is grayish because the neurons are unmyelinated. They don't have myelin sheaths on the axons of the neurons. The white matter, which is inside of the brain, it looks whitish because the axons of those neurons are myelinated. They have a myelin sheath surrounding the axon. This is why they have a different coloration. And guys, take a look here, everybody. You see this structure right here that I just circled for you? This is the corpus callosum right here. And again, the corpus callosum is the structure that is going to bridge, is going to connect the right hemisphere with the left hemisphere. And right here in the middle of the two cerebrums, we are going to find the longitudinal fissure. You can see right there, there's this line all the way down here. This is the longitudinal fissure. All right, guys. So those are the structures of the brain that I want you guys to know. You need to know those structures by name. They will be on the exam. Let's take a closer look now at the PNS, the peripheral nervous system. And the PNS is divided into the cranial nerves and the spinal nerves. Now, what is the difference between the two? The spinal nerves are the nerves that branch off from the spinal cord. The cranial nerves are the nerves that will branch off from the brain itself. And we can see right here on this cartoon, there are 12 different cranial nerves. Okay, the ones that you need to know are in the exam list. Now, what about for the spinal nerves? The ones for the spinal nerves that I want you guys to know, again, they are included in the exam list, but let me go over a few of them here that I find the most important. And that includes the radial nerve, which is a nerve running right next that's going to extend into the radius. We have the ulnar nerve, and we have what is called the median nerve. And the blockage of these nerves here will cause different problems and with the control of our hands. A blockage of the radial nerve is going to cause what is called a wrist drop. The person is going to be unable to hold the wrist up. A blockage of the ulnar nerve is going to cause ulnar nerve palsy, which they're going to be unable to control these last two fingers here, the 4 and 5. And then a block of the median nerve is going to cause median nerve palsy, in which they're going to have a difficult time controlling most of the digits in their hands. Okay. What about in the lower limb? In the lower limb, I want you guys to know the femoral nerve. Again, this is not an artery. It is a nerve, not the femoral artery, but the femoral nerve. And then on the posterior side, I want you guys to know the sciatic, which is you can't miss it, the very thick nerve running the back of the leg, which then when the sciatic runs behind the knee, is going to become the common fibular, and then it's going to continue down into the lower leg, right next to the tibia, and that's going to be called the tibial nerve. So I want you to know the sciatic nerve, the common fibular nerve, and the tibial nerve. Okay, any questions? All right. So the last thing I want to talk about, and then we're going to do our in-lab activity for today. We use in the lab, we use animal models, right? Because we don't have cadavers, going around easy to access. So we use animal models. And to study the brain, most of the students in the labs and universities around the country, we use a sheep brain as a model for the human brain. Now, are they the same? No, they're not the same, but we do share a lot of things in common. Okay, so let's go over some of the structures here of the sheep brain. One of the structures that we can see is this line right here in the middle of the cerebrum, the right and left cerebrum. That is what we call the longitudinal fissure. We again can see how the surface of the cerebrum is all convoluted, and these are the gyri, okay? And there is the division of the lobes. So we're going to find here the frontal lobe, we have the temporal lobe, we have the parietal lobe, and then we have the occipital lobe all the way in the back of the brain. And again, all of these lobes are bilateral. They have the right and we have the left. Let's take a look now underneath an inferior view of the brain, using the sheep brain here. And we can see one of the 12 cranial nerves, the olfactory bulb, right here. And these olfactory bulbs, these are the nerves that allow us to be able to smell. We have those olfactory bulbs as well. And then we can see another cranial nerve right here, which are the optic nerves. And they have been cut. And the optic nerves are the nerves that are going to send information from the retina, from the eye, all the way to the back of the brain in the cerebral cortex. And then the optic nerves, they do cross each other, folks. And the point where they cross right here, this is what we call the optic chiasm. So what is the optic chiasm? The point in which the optic nerves cross one another, going from right to left and left to right. Okay. We can see the pituitary gland right here, which would be hanging from the hypothalamus. And then we can see the pons and the medulla oblongata, which are part of the brain stem of the brain. Now let's cut this brain here in half. Again, a mid-sagittal section. And we can see the cerebrum right here. Underneath the cerebrum, we can see the corpus callosum of the brain, which again makes the connection between the right and the left hemisphere. Right underneath the corpus callosum, we're going to find the lateral ventricles right here. Now you can see how there's a membrane right there. This membrane is called the septum pellucidum. The septum pellucidum is a membrane that separates the first and the second ventricle. So there is a very thin membrane separating the two lateral ventricles, the first and the second, okay? All the way here at the bottom, we can see the optic chiasm, which is the point where the optic nerves are going to cross each other. We can see the hypothalamus, which is part of the diencephalon. We can see the thalamus, the big thalamus right there, again, also part of the diencephalon. And we can see the pineal gland, which is part of the epithalamus, also part of the diencephalon. Okay? And then moving back here, we can see the pons, the brain stem. Sorry, the midbrain would be right here. So which is part of the brain stem. So we have midbrain, we have pons, and we have the medulla oblongata right there. And then right here, we are taking a look at the cerebellum, which, by the way, guys, everybody take a note of this. The cerebellum is bilateral. So we have the right cerebellum, we have the left cerebellum, okay? So right here, we're looking at the left cerebellum. And inside of the cerebellum, there's a really cool structure that kind of look like a tree. Look over here. This is what we call the arbor vitae. And the arbor vitae is also called the tree of life. Okay, and of course, this structure is bilateral because we do have a true cerebellum. We have the right arbor vitae of the right cerebellum. We have the left arbor vitae of the left cerebellum. Any questions, guys? All right, what are we doing today? Very simple, very easy. We only have one Edpuzzle video. And that Edpuzzle video, which you guys have access not only today, but anytime before the exam for you to study, is going to walk you through a model of the human brain. And it's going to point out all these different structures that I just talked to you, I just discussed with you right now. And remember, I'm going to be using pictures of models for the exam. So you need to familiarize yourself with them. After you're done with the Edpuzzle video, you're going to jump on Lamaku and you're going to take the quiz for today. After you're done with the quiz, you're good to go. Okay, let's get started, guys.